What I'm going to speak about today is about cell and animal models of dementia, and in particular actually it's tauopathy, so it's quite a specialist area. And one of the problems we've had in this area is that we just don't have enough good models, particularly animal models, but a lot of the cell models don't really model what we're looking at either. So the approach we took was to make a Knockin mouse, or a kind of a Knockin mouse that expresses a particular fragment of tau that we found in a tauopathy brain. And those mice have been around for a little while now, and we've done quite a few behavioural studies on them. We've done neuropathological analysis, and we've done biochemical analysis as well. That's all very well, but while those mice were being generated, we also generated some cell lines. So these are well, various types, but non-neuronal cell lines and neuronal. And we chose non-neuronal cell lines because we specifically wanted to look at microtubule cytoskeletal aspects of tau, because it's a microtubule binding protein. And we found some quite important changes that when you make cells that have this particular fragment of tau, the cytoskeleton is disrupted. So that's all very well, but then we went on to look at neuronal cells as well. So we've now derived neurons from the tau mice, the tau transgenic mice that we've got. And we've started to look at various aspects of what's going on in those cells. The problem really is understanding, when you have a mouse, what is it in the mechanism, what mechanisms are going wrong that you can actually begin to target. So that's one of the reasons why we went to the neurons, and we found some very important changes. Some of the things we found biochemical changes that match what we see in the mice to a large extent, certainly in terms of tau phosphorylation and in terms of modulation of enzymes that moderate and phosphorylate tau as well. But much more critical metabolic processes seem to be affected. So we've found big decreases in the number of lysosomes, for instance, and also big changes in mitochondria using these cell models. So we think that these are going to provide a way of looking in much more detail at a molecular level what's going on when you overexpress disease-associated fragments of tau in neurons.